Welcome back to Bio 102, lecture number 4, chapter 22, The Origin of Species. My name is Mr. Sparks, and I will be your lecturer for this PowerPoint. In the Galapagos Islands, Darwin discovered plants and animals found nowhere else on Earth, and it was here that he developed his theory of evolution by natural selection, or descent by modification. Modification by descent. Here we see the flightless cormorant, which occurs on the Galapagos Islands, hundreds of miles away from any of the nearest cormorant populations that exist on the mainland. The mainland cormorants are capable of flight, and they must have flown to the Galapagos Islands where, through the absence of predation, the flightless cormorant evolved. Speciation is the process by which species split into two or more species. Speciation explains the features shared between organisms due to inheritance from their recent common ancestor. Speciation forms a conceptual bridge between microevolution and macroevolution. Microevolution consists of changes in allele frequency in a population over time. Macroevolution refers to broad patterns of evolutionary change above the species level. The biological species concept emphasizes reproductive isolation. Species is a Latin word meaning kind or appearance or type. Biologists compare morphology, physiology, biochemistry, and DNA sequences when grouping organisms. This is the process of phylogeny that we learned about in the previous lecture. The biological species concept states that a species is a group of populations whose members have the potential to interbreed in nature and produce viable, fertile offspring. They do not breed successfully with other populations. Gene flow between populations holds the populations together genetically. If there is no gene flow, those populations begin to become distinct, and if they become more distinct over time through the various processes that we will describe in this chapter, then they can become different species. Here are two examples of similarity between different species and diversity within a given species. The first example is the eastern and western meadowlark. They're very similar species that were driven apart by the last major glaciation some 10,000 years ago, and that major glaciation came down in through Wisconsin and down into Ohio, and it caused major ecological differences in the climate. And those populations, the eastern and western meadowlarks, were separated by that glaciation event, and they evolved into two separate species. Now, they maintain their distinct nature by virtue of their call. The song of the eastern meadowlark is different than the western meadowlark, so much so that even when the species encounter each other, which they do periodically because the glaciers have retreated and allowed those ecosystems to come back in contact with one another, but because they're different, they have different songs, they no longer interbreed with one another. And diversity within a species, it's easy to use human beings as an example of this. We're all the genus Homo, species sapiens, and there's much different genetic and phenotypic diversity in between all individuals or populations of the genus Homo sapiens. Okay, here's the eastern-western meadowlark. Remember what I said about the glaciation event causing the distinct speciation. Eastern meadowlark, western meadowlark. And here are the various, there's many different types of human beings than just what's illustrated here. I think we're all aware there are hundreds of ethnicities, all emanating from various countries and populations around the world. Reproductive isolation is the existence of biological barriers that impede two species from producing viable, fertile offspring. Hybrids are the offspring of crosses between different species. Reproductive isolation can be classified by whether barriers act before or after fertilization. Now here's a chart that shows on one page pre-zygotic barriers compared with post-zygotic barriers. Okay, pre-zygotic barriers are those barriers that prevent fertilization, and post-zygotic barriers are those that act after fertilization. When the organism is hatched or born, it may experience reduced viability. So here we have different examples of habitat isolation. This is a garter snake, Thanopsis species, which occurs in the in Canada, and there are one occurs upland regions, and the other one occurs in wet marshy habitats. And because they both have these habitat specialties, they wind up becoming reproductively isolated, and that could cause speciation over time. Here we have the eastern and western spotted skunk. They breed during different times of year. So even though they might produce offspring, they never do, because the breeding season is distinct for each species. And here we have the blue-footed booby, which is depends on certain dance maneuvers, if you will, in order to woo its prospective mate, and then that will occur only if they have the proper behavior. So that is a type of behavioral isolation. Then we can talk about mechanical isolation. Different species, like a chicken, cannot mate with a dog. There's mechanical isolation there. They're much too different. Here is an example of closely related species of these snails, the genus Bradythena, which one snail has a shell that spirals in the right-hand direction. The other snail has a shell which spirals in the left-hand direction. And because these are different, they are unable to line up their gonadopores, their reproductive organs, and so they wind up being distinct. Now, it's interesting to note that the direction of the spiral of the shell is controlled by one single gene. So that single gene, if it can flip during the development of the egg, then it can cause these two populations to become distinct. Okay, gametic isolation is like what happens. It can happen between any species that are dissimilar. In this case, it shows two different types of sea urchins, and they spawn at the same time. They have the same mechanical mechanism. but the gametes do not produce do not fertilize. So the gametic isolation prevents fertilization. Now, after fertilization has occurred, you can have offspring. But are these offspring viable, and are they fertile? Well, that is the question that could lead to speciation. So reduced hybrid viability is when, for example, some species, some salamanders of the genus Ensatina in California occur in different species, and those species may overlap, and they may produce offspring, but those offspring may be they may not be strong enough to survive until reproductive age. Reduced hybrid fertility occurs in the mule, which is an offspring of the donkey, a male donkey, and a female horse. It is a vigorous hybrid that is capable of many feats of strength, but it has reduced fertility. The mule is essentially is not fertile. So that is another mechanism of postzygotic barrier. And then we can also talk about hybrid breakdown, which this is more complex, and it occurs in plants generally. In let's see, what's the word I'm looking for? In polyploid plants that develop genetic. We'll talk about this more later, but some plants develop problems with their called a non-disjunction event, which means that the gametes have more chromosomes than they normally would, and those sometimes can produce hybrid viable hybrids that produce offspring. So viable fertile hybrids can be produced by allopolyploidy in the different plants. The example here is shown is rice. Okay, so those may produce offspring in 1 or 2 generations, but by 3 or 4 generations, the fertility breaks down and the plant can no longer survive. It can no longer produce a viable fertile offspring. Okay, pre-zygotic barriers block fertilization from occurring by impeding different species from attempting to mate, preventing the successful completion of mating, and hindering fertilization if mating is successful. Okay, pre-zygotic barriers. Before the mating attempt, 2 different habitat isolation in the garter snake, temporal isolation in the spotted skunk, and behavioral isolation in the blue-footed booby. Habitat isolation, 2 species encounter each other rarely or not at all because they occupy different habitats, even though this not isolated by physical barriers. Okay, here's the example of the garter snake. Here's the aquatic garter snake and the terrestrial garter snake. Temporal isolation, species that breed at different times of day, different seasons, or different years, and cannot therefore mix their gametes. The western spotted skunk and the eastern spotted skunk, their breeding season for the western spotted skunk is in the spring, and for the eastern spotted skunk it's in the fall. So they never go into estrus around each other. They may occur in the same habitats, but they're never in their breeding season at the same time. Behavioral isolation, courtship rituals, and other behaviors unique to a species are effective barriers. Okay, the blue-footed booby will only mate with other blue-footed boobies. It has a special dance that it does, a ritual prior to mating, that the male must appease the female, and in so doing, no other species are going to be able to breed with the female blue-footed booby. Okay, pre-zygotic barriers continued. Include mechanical isolation and gametic isolation. The two snails cannot mate because of the differences in their shells, and the sea urchins, their gametes will not fertilize one another, so the fertilization does not occur. Mechanical isolation, morphological differences prevent successful mating. You can look at these snails, or like a chicken and a dog, or any species, a lion and a cat, a house cat. They're not going to be able to mate, so they've evolved into distinct species. Okay, here's the gonadopores. They can't line them up right, therefore they eventually leads to speciation. Gametic isolation, the sperm of one species may not be able to fertilize the eggs of another species. Here are the two different species of sea urchins. Even though they reproduce at the same time, they overcome temporal isolation, and they have the same mechanical adaptations for fertilization. They cannot actually fertilize one another's eggs, and sperm cannot fertilize the eggs because of biological differences between the two. Post-zygotic barriers prevent the hybrid zygote from developing into a viable, fertile adult by reducing hybrid viability, reducing hybrid fertility, and hybrid breakdown. Okay, so here's our post-zygotic barriers. Reduced hybrid viability is the Encetina salamander. A hybrid from two different species can occur. It can be born and live, but it may, in this case, its coloration is skewed so that it causes it to be conspicuous to predators. And for other reasons, it may be also less viable, less able to survive in the environment. Reduced hybrid fertility. The donkey, the male donkey and the female horse produces the viable mule, which is an incredibly strong and useful animal, draft animal, but it's not fertile. It cannot produce. A mule can't breed with another mule, and a mule can't breed with a horse, and a mule can't breed with a donkey. It's become an evolutionary dead end. And then hybrid breakdown occurs primarily with plants because of allopolyploidy or polyploidy, which is which occurs through non-disjunction events in the gametes of the plant species, and they may produce a viable, fertile offspring that produces one or two or three or four or five generations of viable, fertile offspring. But as the generations go forward, each generation is less and less viable until finally that offspring is an evolutionary dead end. It can no longer reproduce. That's hybrid breakdown. Reduced hybrid viability. Genes of the different parent species may interact and impair the hybrid's development or survival. Okay, here's Encetina hybrid. There it is. It's conspicuous to predators. It has other factors that reduce its viability. Reduced hybrid fertility. Even if hybrids are vigorous, they may be sterile. Okay, donkey, horse, mule, sterile. Evolutionary dead end. Hybrid breakdown. Some first-generation hybrids are fertile, but when they mate with another species or with either parent species, offspring of the next generation are feeble or sterile. Okay, here's the example of the rice. Okay, so I guess that's supposed to be the large one is the first generation, then they get smaller and smaller over time. They're no longer viable. They break down. That's hybrid breakdown. Okay, here's the summary: habitat isolation, temporal isolation, behavioral isolation, all occur before the mating attempt. Mechanical isolation and gametic isolation all occur between the mating attempt and fertilization. After fertilization, there's reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. Limitations of the biological species concept. The biological species concept cannot be applied to fossils or asexual organisms, including prokaryotes. So you don't know what the breeding relationships were of certain dinosaurs. You have to use the morphological species concept when you're dealing with fossils, basically, because you don't know what their relationships were. And asexual organisms, you have to look at their genetics and morphological characteristics, like, for example, of the bacteria and the archaea, in order to determine what species they may fall in, what taxonomic category. So it's hard to know. The biological species concept emphasizes the absence of G-flow. So if biological species means that two populations are becoming distinct species if there is no longer gene flow. However, gene flow can occur between distinct species. For example, grizzly bears and polar bears can mate and produce growler bears. And this has occurred in the wild a couple of times. In captivity, it's possible to breed lions and tigers and produce what's known as either a liger or a tyon. And those are hybrid organisms, hybrid animals, but they are not fertile. Growler bears are relatively new. I'm not sure if it's known whether they're fertile or not, because there's something that occurring with greater frequency as a result of climate change and global warming. The absence of sea ice has caused the polar bears to spend more time on the land, where they're more likely to encounter the grizzly bears. So here's the grizzly bear and the polar bear. They're obviously distinct species, or so they appear to be, but then they can unite and breed and form the hybrid growler bear. There are other definitions of species. Other species concepts emphasize the unity within a species rather than the separateness or different species. The morphological species concept defines a species by structural features. It applies to sexual and asexual species, but relies on subjective criteria. Nowadays, it's particularly dependent upon genetic information. The ecological species concept views a species in terms of its ecological niche. It applies to sexual and asexual species and emphasizes the role of disruptive selection. The phylogenetic species concept defines a species as the smallest group of individuals on a phylogenetic tree. It applies to sexual and asexual species, but it can be difficult to determine the degree of difference required for separate species. Speciation can take place with or without geographic separation. Speciation occurs in two ways. There's allopatric speciation and sympatric speciation. Patria means land, and allo means different, and sim means the same. So allopatric speciation means speciation that occurs in different lands. Sympatric speciation means speciation that occurs in the same lands. Allopatric speciation forms a new species while geographically isolated. So here's an example of an aquatic system separated by a barrier that could occur, that could present itself during drought. Here's the drought event. It separates these two populations. This is allopatric speciation. One speciation becomes more adapted to its environment, and it becomes distinct from the parent species. And sympatric speciation is a subset of species. A subset forms a new species without geographic separation. So there could be some other natural selective activity influencing these populations and cause speciation to occur within that same territory, within the same population. Generally, sympatric speciation occurs in plants for the reason that I identified with you earlier when we were talking about polyploidy speciation with the rice plants. For some reason, plants are much more susceptible to polyploidy, and therefore that polyploidy event we'll discuss, it changes the chromosomes of the organism, and if that organism is fertile, then it can become distinct from its parent species. That's normally what happens in sympatric speciation, and it occurs in certain tree frogs, certain aquatic frogs, different amphibians. It's more likely to occur in them, and it's very rare in mammalian species. Allopatric speciation is gene flow is interrupted when population is divided into geographically isolated populations. For example, the flightless cormorant of the Galapagos likely originated from a flying species on the mainland. So that flying, flightless cormorant flew to the Galapagos Islands, and there it became distinct. It found enough resources to survive. There was never any other communication besides that original event, and it became distinct. It evolved separately through allopatric speciation. The definition of a geographic barrier depends on the ability of the population to disperse. For example, a canyon may create a barrier for small rodents, but not for birds or coyotes or pollen. Separate populations may evolve independently through mutation, natural selection, and genetic drift. Reproductive isolation may arise as a result of genetic divergence. For example, mosquito fish in the Bahamas comprise several isolated populations in different ponds. In some of those ponds, they have high predation, and in other ponds, they have low predation. Under high predation, the body shape enables rapid bursts of speed, and under low predation, they're less concerned about predation, so a body shape favors a longer, steadier swimming body. So under the selective pressure of predation, you can see the difference between these mosquito fish. Now, if this continues over time and those populations continue to be isolated and there is no predation in the B population, then the A population and the B population could become distinct. That's allopatric speciation. Evidence of allopatric speciation. 15 pairs of sister species of snapping shrimp, Alphaeus, are separated by the Isthmus of Panama. These species originated from 9 to 3 million years ago when the Isthmus of Panama formed and separated the Atlantic and Pacific waters. Okay, so here we've got two pairs of these species. Remember, they talk about 15 pairs, but these species are all very morphologically similar, but they're separated by the Isthmus of Panama, which arose from volcanic events about 9 to 3 million years ago. When the Isthmus of Panama arose, it then caused these species to become distinct through allopatric speciation. Now, it's interesting to note that that these shrimp, sometimes called pistol shrimp, are capable of stunning their prey. They clap their claws together, and they do it so quickly that it causes a small bubble to originate in the water, and that the amount of energy causes an intense amount of heat as well, and it can stun prey items that are within close enough range. So the shrimp doesn't actually touch its prey, it just claps its jaws shut, causing a cavitation bubble. The cavitation bubble collapses, and this causes a physical force in the water, and that stuns the prey, and then the shrimp goes out and consumes the prey item. Here they are. You can see that pistol shrimp claw right there. Now see how similar they are. Okay, nutting eye and Mill's eye are what we're looking at. I don't know why they have it in this area, but here's A. formosus. It's related to A. panamensis. And here's A. nutting eye. It's related to A. milsae. Regions with many geographic barriers typically have more species than do regions with fewer barriers. So, for example, in the Papua New Guinea, which is an extremely mountainous area, which has tons of mountains and valleys, each of those major valleys can have different birds. These birds are known as the birds of paradise, and they're quite spectacular animals, and they're highly distinct from one another, and that's because of the geographic barriers between those areas. Reproductive isolation between populations generally increases as geographic distance between them increases. We're going to see a little graph about this at the end of the chapter. I don't know why they put it there and they didn't put it here, but that's where it is. Barriers to reproduction are intrinsic. Separation itself is not a biological barrier. There could be separation between two populations, but if the environmental conditions are the same and consistent between those two populations, then there will be no cause for evolution, no cause for natural selection if the environmental conditions remain the same. Intrinsic reproductive barriers can develop in experimentally isolated populations. Okay, here's the fruit flies. There's an initial population of fruit flies. They're all the same here. They're divided out. Some are raised on starch medium. Some are raised on maltose medium. And then they mix the two generations after 40 generations. And you can see here that the starch flies tend to mate with the starch flies, and the maltose flies tend to mate with the maltose flies. And the starch flies and the maltose flies seem to avoid one another, and the same goes with the maltose flies and the starch flies. They seem to avoid one another. So this indicates a potential for speciation here through allopatric speciation based on the different mediums that the fruit flies are raised in. Now here we see what different populations, males and females, from the starch population, and there's basically very little difference here. The matings in the control group are pretty much similar. They found the same to be the case for the maltose group. So this says that this mating between the starch and the maltose and the maltose indicates a potential speciation event. It's not really a speciation because we're talking about fruit flies here, but if you raise them over 1,000 generations or so, it could cause a difference in speciation. Okay, in sympatric speciation, speciation takes place in the populations that live in the same geographic area. Sympatric speciation occurs when gene flow is reduced in groups that remain in contact through factors including polyploidy, habitat differentiation, and sexual selection. Polyploidy is the presence of extra sets of chromosomes due to accidents during cell division. Polyploidy is much more common in plants than in animals. An autopolyploid is an individual with more than 2 chromosome sets derived from one species. Offspring of matings between autopolyploids and diploids have reduced fertility. Okay, here's a diploid and has a cell division error leading to a tetraploid cell. During meiosis leads to a tetraploid cell, and then that produces a 4N gamete. These 2N diploid cells can join with this, can join together, and they might produce this tetraploid cell. An allopolyploid is a species with multiple sets of chromosomes derived from different species. Allopolyploids cannot interbreed with either parent species. Okay, so here is species A is allopolyploid. A meiotic error. Chromosome number does not reduce from 2N to N. So the unreduced gamete has 4 chromosomes, and the normal gamete has 3 chromosomes. And you can't join these two together and produce viable offspring because they'll have 7 chromosomes. It has to be an even number to produce a viable offspring. Okay, so hybrid with 7 chromosomes, not viable. Okay, a normal gamete again joins when the unreduced gamete has 7 chromosomes. A new species, viable fertile species, an allopolyploid, has 10 chromosomes. So it's diploid 10 chromosomes, and then it becomes viable. Many important crops, oats, cotton, potatoes, tobacco, and wheat are polyploids. In fact, scientists speculate that as much as 80% of plant species are polyploids, or originated with polyploidy. Sympatric speciation can also result from the appearance of new ecological niches. For example, the North American maggot fly can live on native hawthorn trees as well as more recently introduced apple trees. And the ones that survive on the native hawthorn trees become ecologically distinct, just like the fruit flies did in the maltose experiment, sucrose maltose experiment. The starch maltose experiment. Sexual selection can drive sympatric speciation. Sexual selection for mates of different colors has likely contributed to speciation in cichlid fish in Lake Victoria, Africa. This is a very interesting phenomenon here. Okay, under normal light conditions, the female of the species will select only within their species. But under changed light conditions, under monochromatic orange light, where these species look similar, the female will mate with either species and produce viable offspring. So this distinction of speciation suggests that the speciation is a relatively recent event. Okay, here's normal light, cichlid, monochromatic light. Okay, so they look different under normal light, and they look pretty much the same under monochromatic light. So allopatric and sympatric speciation. In allopatric speciation, geographic isolation restricts gene flow between populations. Reproductive isolation may then arise by natural selection, genetic drift, or sexual selection in the isolated populations. Even if contact is restored between populations, interbreeding is prevented by reproductive barriers. In sympatric speciation, a reproductive barrier isolates a subset of a population without geographic separation from the parent species. Sympatric speciation can result from polyploidy, natural selection, or sexual selection. A hybrid zone is a region in which members of the different species mate and produce hybrids. Hybrids are the result of mating between species within incomplete reproductive barriers. A hybrid zone can occur in a single band where adjacent species meet. For example, two species of toads in the genus Bombina, the fire-bellied toad, interbreed in a long and narrow hybrid zone. So here are these. The yellow is the yellow-bellied toad habitat, and the orange is the fire-bellied toad range. They do encounter one another along this narrow band. That narrow band is where hybrids are formed, and that is called the hybrid zone. Okay, so here is the frequency of a particular allele, which is common to the yellow-bellied toad, and it over the course of the hybrid zone it decreases, and the fire-bellied toad zone you have almost negligible amount of that allele. Okay, here's the hybrid zone right here through the Caucasus Mountains here. Hybrids often have reduced fitness compared to parent species. The distribution of hybrid zones can be more complex if parent species are found in patches within the same region. When closely related species meet in a hybrid zone, there are three possible outcomes: reinforcement, fusion, and stability. Okay, let's just click through here. All right, here is population. It has gene flow going between the multiple populations. Each one of those blue splotches represents a population. Those populations begin to diverge, perhaps through a allopatric event, a mountains rising, or some event that causes the distinction between the two populations. Then here is the hybrid zone, and there are the hybrid individuals in that hybrid zone. Okay, there are the possible outcomes: either reinforcement, in which the two populations continue to join together, and they produce a third hybrid population; fusion, in which those populations eventually join back together on the other end of that event, that allopatric event. Or stability, in which case that hybrid zone just continues on throughout time. It seems that the hybrid zone for the fire-bellied toad is a pretty stable hybrid zone. Reinforcement occurs when hybrids are less fit than in the parent species. Natural selection strengthens or reinforces the reproductive barriers, and over time, the rate of hybridization decreases. Where reinforcement occurs, reproductive barriers should be stronger for sympatric than for allopatric species. Fusion of the hybrid of the parent species into a single species may occur if hybrids are as fit as parents, allowing substantial gene flow between species. For example, researchers think that pollution in Lake Victoria has reduced the ability of the female cichlids to distinguish males of different species. So they are, because of environmental conditions causing increased turbidity, which is suspended particles in the water, it's changing the way light affects the coloration of the different species, and they're looking more like the same species, and so that what could have become a speciation event is now becoming a fusion event. This might be causing the fusion of many species. Okay, remember we talked about the cichlids before, and with the orange light. Well, the orange light was just mimicking the turbidity. So here you can see a hybrid that has occurred from these two different species here, hybridizing and then fusing into a viable offspring. These populations will become less distinct. Stability of the hybrid zone may be achieved if extensive gene flow from outside the hybrid zone can overwhelm selection for increased reproductive isolation inside the hybrid zone. In a stable hybrid zone, hybrids continue to be produced over time. Speciation can occur rapidly or slowly and can result from changes in a few or many genes. Many questions remain concerning how long it takes for species to form or how many genes need to differ between species. Broad patterns in speciation can be studied using the fossil record, morphological data, or molecular data. The fossil record includes examples of species that appear suddenly, persist essentially unchanged for some time, and then apparently disappear. These periods of apparent stasis punctuated by sudden change are called punctuated equilibrium. This is the predominant theory of evolution at this point in time. The punctuated equilibrium model contrasts with the model of gradual change in a species' existence. So here we show some drastic environmental change occurs, and this is over thousands of generations, but it's a relatively short period of time in geological terms. And then which makes these species distinct, and then these species continue as distinct throughout time. Gradualism suggests that these environmental changes occur more gradually over time, and that speciation occurs over longer periods of time. In fact, punctuated equilibrium dictates that both of these models can occur, although not simultaneously. But a speciation event can be punctuated, or a speciation event can be gradual. The punctuated pattern in the fossil record and the evidence from lab studies suggests that speciation can be rapid. For example, the speciation of the sunflower Helianthus anomalus, originated from the hybridization of two other sunflower species and quickly diverged into a new species. Okay, so these two different sunflowers joined together. They produced an experimental hybrid with this, a polyploid hybrid. The chromosomes of those of the polyploid hybrid became more and more similar to the original population over time, and allowed for those. Over time, the chromosomes in the population of the experimental hybrids became similar to the chromosomes of H. anomalus individuals from natural populations, and this suggests that the observed rise in the fertility of the experimental hybrids may have occurred as selection eliminated regions of DNA from the parent species that were not compatible with one another. Overall, it appeared that the initial steps of speciation process occurred rapidly and could be mimicked in a laboratory experiment. The interval between speciation events can range from 4,000 years in some cichlids to 40 million years in some beetles. It all depends on environmental conditions and the fertility of that organism. You might expect speciation to occur more frequently in prokaryotes that reproduce more rapidly, or insects over time may have a more rapid speciation. But for solid speciation events to occur across all plants and animals, the average is about 6.5 million years. So it takes a considerable amount of time for species to become distinct. A fundamental question of evolutionary biology persists. How many genes change when a new species forms? Depending on the species in question, a speciation might require the change of only a single allele or many alleles. For example, in Japanese euhedra snails, the direction of the shell spiral affects its mating and may be controlled by a single gene. Remember, we talked about this earlier with the Bradybania snails. Similar gene. It's just a single gene that can change in that organism during meiosis or during the development of the snail, and it can change. It can affect the direction of the shell spiral, and that affects its ability to mate. In monkey flowers, two loci affect flower color, which influences pollinator preference. Pollination, which is dominated by either hummingbirds or bees, can lead to reproductive isolation of the flowers. In other species, speciation can be influenced by larger numbers of genes and gene interactions. So here's the monkey flower. Its coloration is affected by two loci on a given gene. The red flowers are preferred by the hummingbirds, and the pink flowers are preferred by the bees. So you can cross these and make a pink flower more reddish, and that will make it more attractive to both hummingbirds and bees. And you can take a red flower, which is attractive to hummingbirds, and make it pinkish, which makes it attractive to bees and hummingbirds. So these two flowers are distinct by virtue of their pollinators, but you can change which pollinators are attracted to them by transferring the alleles for flower coloration. Macroevolution is the cumulative effect of many speciation and extinction events. Okay, so we talked about microevolution, which is the change of these alleles in a population that might make it distinct from another population, but within the same species. But those species can become distinct, and then even further than species over time, genus and family, class and order, they can all become, those differences can accumulate over time and create macroevolution. Okay, here's that chart they talked about earlier. I don't know why they put it here at the end, but here's a geographic distance and the reproductive isolation value. Okay, this is relatively close here, and this is this could be kilometers, and here's the distance from the next nearest population is much further away, and it has a greater reproductive isolation value. So closer, lower reproductive isolation value, further away, greater reproductive isolation value. Okay, here's our allopatric speciation and sympatric speciation. Allopatric are when those populations are separated by geographic or temporal barriers. Sympatric speciation are when speciation occurs when those populations are in contact with one another. Okay, here is polyploidy in wheat, and thankfully polyploidy does occur in plant species, because if you look at these original wheat species, these Triticum monocossum is the original wheat that primitive people began to consume. And then it crossed with wild Triticum and wild Triticum talshii, and this cross produced a more viable, it was a more attractive wheat, and this is the wheat that becomes bread wheat. So because of polyploidy in these species, here's the original polyploidy, original diploid species here, combining to join to create the diploid 42 chromosomes here. That creates the bread wheat.